All right, let's go ahead and get started. Thank you for joining the team at Hands-On Labs today for our discipline-focused live demo series. Today's topic is dedicated to HOL's biology online curriculum and kits. Before I hand the mic over to our host, let me take a moment to review the webinar logistics and give a brief introduction on today's event. We encourage our attendees to participate in the conversation by asking questions and sharing resources. Please use the chat function to join in on the conversation. Be sure to toggle the window to all panelists and attendees if you'd like the entire group to see your comment or question. If you have any questions, add those in the Q&A box. You can find this section in your toolbar. We have several distance learning specialists on the panel today to help answer any questions you have in real time. For questions pertaining to kit cost and customization, please work directly with your dedicated distance learning specialist once the webinar concludes. Dr. Duane will address any remaining questions at the end of the demo so you have clarification on important topics. We'll start the presentation with a quick poll to gauge our audience temperature on their past experience with online teaching. To participate in the polling segment, submit your response when you're prompted with the poll pop-up. At any point in the conversation, you can raise your virtual hand and our distance learning specialist will send you a private chat message to support your needs. All attendees will be entered into a raffle to win a $100 Amazon gift card. We'll share the lucky winner in our follow-up email at the end of the day, along with a recording of this webinar for you to reference moving forward. So what can you expect to see today? During our biology live demo, our chief resident scientist, Dr. Duane Cagle, will help you investigate biological phenomena with HOL's rigorous online curriculum and the powerful platform that hosts the content from both an instructor and student perspective. Before I introduce our host and distance learning specialist, let's get an idea of our audience's proficiency with online lab learning by launching the poll now. All right, it looks like we have a nice mix between instructors who have done this in the past and those who are new to distance learning labs. Thanks for participating. If you don't already know our host today, Dr. Duane Cagle is our chief resident scientist and has been the brains behind Hands-On Labs curriculum development for the past 9 years. We also have several HOL distance learning specialists on the call today who will be manning the chat and providing helpful resources throughout the webinar. A warm welcome to Pam Paisawang, Justin Johnson, Eric Kenyon, and our main host, Dr. Duane Cagle.
Well, great, Justin. Thank you so much for the warm welcome. And more importantly, I want to thank all the audience for attending today. We are very excited to have you join us in this webinar and learn more about options for teaching your biology course, laboratory online. So I want to start today by letting you know what to expect. We're going to spend a fair amount of time talking about our curriculum that is used to support online biology laboratories, and an equal amount of time introducing you to our lab manual digital course platform called HOL Cloud. It is indeed a cloud-based virtual learning platform that coincides with our lab kits, which contain physical materials for students to conduct hands-on procedures and gain the competencies that are required of them based on your particular courses. So what are we seeing on the screen right now? Well, what we're looking at is the landing page of our digital lab manual. And this is going to be the student view, and that's how we're going to begin our session today, is experiencing our lab manual curriculum and getting an idea of the physical materials in our kit, just as a student would use these materials in their online biology lab course. So on the landing page for the lab manual, the student first encounters any notes from their instructor that we see in the brown box at the top of the page, immediately followed by their course prerequisites. Now, I know one of your main concerns when conceptualizing taking your laboratory course online is lab safety and the other requirements that are expected of your students before they begin laboratory experiments in your on-campus setting. So we have accommodated all those needs within the prerequisites that are included in each of our lab manuals. In this particular biology course, you'll see that the prerequisites consist of a getting started lesson, a lab safety lesson, and lastly, a using the V-scope. And while we're not going to take the time to look at any three of those right now in any amount of detail, I just want you to realize that we configured our lab manual in a manner such that students must complete any and all prerequisites before gaining access to the lab curriculum proper. Meaning a student has to first complete getting started, which familiarizes them with all the technology in this cloud-based lab manual platform, so the students can gain confidence and competency in answering interactive questions, uploading data, and then submitting their final lab reports. Likewise, in the lab safety lesson, students are required to review an array of safety information that pertains directly to the types of physical materials received in their kits. They're also expected to answer a series of questions on how to handle various situations that they might encounter when conducting experiments. And then the last part of the lab safety lesson is that students do sign a safety agreement, which in essence, the student acknowledges that they will comply with all of our procedures and use our materials in the way that they were designed. And then in payment for that, as a business, we provide your students with liability coverage should they incur any accident while conducting procedures using our physical materials. And then lastly, this being a biology course, I know that a lot of our institutions have a requirement for some type of microscopy. As a company, we provide options for that. We provide physical microscopes. We provide curriculum that requires students to display competency in using the microscope. And then lastly, as an alternative, we also have an interactive virtual microscope that we call the V-scope. For instructors who opt for their students to use the virtual version of the microscope, we have included a prerequisite training lab for that technology. If we have enough interest, by the end of today's session, we'll take a look at the V-scope. I know in some of these webinars, we have a lot of interest for it, and others not so much. It really is discipline specific. But if you would like to see that, make a note in the chat as we proceed through the demo in the next 45 minutes or so. And given we have time, I'll give you a quick tour of the V-scope at the end. So without further ado, let's take a look at our curriculum and how students use it to conduct hands-on procedures in the biological sciences. Now, first and foremost, as you're looking at these 5 lab titles, you're probably thinking this spans quite an array of the biological sciences, and this must be a very unique course, or conversely, a very incomplete course. And indeed, this is set up strictly for demo purposes. This is not a live actual course. So rest assured that our biology library is very complete. In a typical course, would consist of a dozen or so lessons. So again, these titles are for example purposes only. But as a student, when we click on a title, we see that it opens into a series of sections. And these sections begin with terms such as exploration, various exercises, and they culminate in an evaluation section. Now, each of these sections is gated, meaning a student has to complete them in order. So being a student, let's start with exploration in this enzymes lab. Now, you'll notice on the landing page for the exploration section, I also see the titles of the two upcoming sections for this particular lab. And again, all of these titles begin with the letter E: exploration, experimentation, and evaluation. Now, if you have a background in teaching specifically as opposed to biological sciences, you may recognize these terms as representing 3 of the 5 E's of science learning. And this all fits into a much larger pedagogical approach to teaching called constructivist learning theory. In a nutshell, it just means that students learn new information based on the foundation of knowledge that they already have. So it's a building or a constructive process. So in the exploration section, we want to provide a student with all of the background content and concepts review that they need to fully understand the procedures that they'll be conducting in the upcoming section, the experimentation. And then lastly, in the evaluation section, we want the student to have an opportunity to demonstrate everything they've learned about the lab topic for that particular week. So in this case, all about enzymes. And we'll see examples of each of these sections in detail in just a moment. Now, you'll notice that the next thing a student encounters in the lab manual after opening a particular section is a time allocation bar in gray in this particular instance. And not only does it tell them the time allocation for the section that they've just opened, but for the upcoming sections also. And indeed, time allocation is a remote science student's main, or one of their main challenges to being successful in an online laboratory course. You know, this is a very different world for your students compared to what they may be used to, in that going to campus for 1 to 3 days a week for, 1 or more hours each day to conduct laboratory procedures. In an online environment, it is solely up to your student to set aside the requisite amount of time to be successful with their assignments. So it's imperative that we provide each and every student with as much notice as possible of how much time they need to allocate to be successful. Following the time allocation statement are clearly defined learning objectives. Now, again, if you have a background in teaching theory, you may recognize how these learning objectives are written and the fact that they conform to Bloom's taxonomy. Bloom's taxonomy fits into constructivist learning theory. In a nutshell, it simply states that there are different levels of comprehension, and those comprehension are connotated by particular verbs, and that learning objectives begin with those specific verbs. So if we're talking about learning objectives for the exploration section, the exploration section is a conceptual review of the content. So the learning objectives are going to have verbs that are much more lower level of learning. So things such as define and describe and discuss. Now we'll see the verbiage become a lot more active and hands-on oriented and at a higher level in the upcoming sections. But once a student has been notified of what they're expected to achieve in a particular section, in this case the exploration, they are going to follow that up with taking a pre-test exam or group of activities, if you will, labeled Test Your Knowledge. Now, Test Your Knowledge activities are specifically designed to assess the student's inbound knowledge of the subject matter that will be covered in the experiment. These Test Your Knowledge activities are always designed as drag and drop. Drag and drop activities have been shown in studies of learning theory to be the most engaging type of activities new students can have when initially interacting with digitized curriculum. So So it's imperative that we engage your students as much as absolutely possible in the first activity that they perform within the Lab Manual platform. Now, if this were a live student instance as opposed to this demo, I could actually move these answers. So I could drag and then drop them in the appropriate cells. Now, this is a matching activity. And when a student completes that in real time and hits next, they're actually provided with feedback. And I've got a screenshot I can show you of what that feedback looked like for this question. So if we had dragged and dropped our answers, this particular student got 2 out of the 5 correct. But the important part here is they are provided with immediate feedback so they can quickly identify any knowledge gaps that they have coming into this lab. Again, online laboratory students are at somewhat of a disadvantage, if you will, or an advantage, depending on how you look at it, compared to their on-campus peers. You know, your on-campus students are almost always conducting laboratory activities after you have covered the topics in the lecture section of your course. That is not always the case for your online students. You may find them frequently working ahead of your lecture due to their time constraints and how they have to budget their time allocations for all of their course activities. So in those instances, it's imperative that we make the student aware if they are lacking sufficient inbound knowledge to be successful in this lab. And once they've identified any of those knowledge gaps that they have, we are going to provide them with sufficient content so they can be successful in understanding their experimental outcomes. Now, again, these activities are always drag and drop. They could be matching like we just saw. They could be categorization type questions, i.e. true, false categories like we see here. They can also be image labeling. You know, if one of the activities your student's going to be expected to understand coming in is the various components of a diagram, let's say if we're talking about photosynthesis, for example, you know, a typical photosynthesis diagram where all the labels would fall, then we're going to include a diagram in these test your knowledge questions and give the student an opportunity to label that to assess their inbound knowledge. Now, once a student has completed the test your knowledge activities, they do encounter the lab manual proper for the exploration section. And each exploration page is designed very, very purposefully. And it always consists of one or more paragraphs of text immediately followed and reinforced by a visual representation of those concepts. So here we see an example of one paragraph of text with important terms in bold. Those terms are being defined in those particular sentences, followed by a graph that represents the content that's provided in that paragraph. It is very important to visually engage students as they're reading content. Further down the page, we'll see another example of that with a diagram of enzyme activity. And then lastly, many of our content pages also include did you know boxes. And these boxes are designed to allow a student to relate the new content that is being covered on the page with concepts they should already be familiar with. And from a learning theory standpoint, we know that anytime a learner can relate to new information, they do comprehend it more efficiently. Now, at the bottom of each exploration content page, there is another activity for the student, this time labeled question time. And a question time activity is always a multiple choice or true-false question where a student selects the appropriate answer that is always related to the content that has just been covered on that particular page. Again, it can be true-false, as we see here, or it can be multiple choice, as we see in question 2. But again, it's designed for a student to confirm that they understand the content that has been presented on the page. They can't advance to a new page of content until they've answered all of these question time activities at the bottom of each page. Now, students are also provided feedback in these instances so they can again identify any knowledge gaps they may still have. And we can see an example of that here to a multiple choice question where the correct answer gets a check mark. If the student answered incorrectly, it's highlighted in red with an X, and the students provided with feedback with what the correct answer is and how it is included in the original question. Now, once a student submits all questions per page, they automatically advance to the next content page. However, should they have missed one or more of those questions, they can always return to the original page where the questions occurred and review the content. So all is not lost. And our goal in designing a lab manual is to assure that the student understands all important information before beginning the hands-on procedures. Now, most exploration sections consist of multiple content pages. In this particular example, we see there are 3 of those content pages. And again, at the bottom of each page, there are question time activities. Again, pages are always accompanied by graphic representations of the content. And then many pages also have these did you know boxes that allow students to relate the content that's being covered with ideas they already understand. Now, once a student completes the exploration section, the experimentation section is then unlocked. Now, the experimentation section, as you might imagine, is the section of the lab manual that contains the step-by-step procedures. But before a student begins those procedures, it is important, again, that they are reminded of the amount of time they need to set aside to be successful. Not surprisingly, the time allocations for the experimentation section are longer than those for either the exploration or evaluation sections. As promised, the learning objectives associated with the experimentation section are more active by nature and connotate a higher level of comprehension. So here we see terms such as demonstrate, analyze, and relate. Page 2 of every experimentation section is a materials table, and we divide the materials table into 2 sections. The first section are materials that students supply from their homes, and the second section, very importantly, are the physical materials that we provide within the lab kit that will be used for the upcoming procedures. Again, student-provided materials are typical household items such as matches or a lighter. In this case, students need to freeze something before they use it. Cleanup materials such as paper towels and water. And then in this particular experiment, because it's dealing with enzymes, the student's using a couple common food items such as milk and granulated sugar. Now, what we provide in lab kits is laboratory equipment as opposed to common household items. So we're going to provide things like burner fuel and burner stands, digital precision scales that measure to 1/100 of a gram, glassware, indicator strips of various types, test tubes, test tube racks, other types of lab equipment such as weigh belts and well plates, and then also chemicals. In most of our experiments, the chemicals come separate inside the lab kit. They're secured in plastic bags, and then the chemicals themselves are often packaged in sealed pipettes that are single use, and we'll see examples of those in the upcoming procedures. You'll notice the volumes of the chemicals that we provide students are very, very small. So in each of these examples, it's only 1 milliliter. I know one concern, aside from student safety, that you probably have being an educator considering taking your lab course online, is how do students safely dispose of any materials they're using for their experiments. Again, we provide students with very small quantities of materials that should pose minimal, if any, disposal concerns for the student and the location that they live. So once a student has retrieved everything from their kit, they're ready to begin the procedures. And we design our procedures very, very purposefully. You'll notice the steps are very well defined and clearly written, and that those steps are always accompanied by a series of photographs of how the student is to be using those materials. Challenge number 1 for your online remote science students is they cannot get immediate answers to any questions they have, unlike their on-campus peers. You know, your on-campus students can raise their hand and you or the lab TA can walk over and answer a question easily for the student, or the student can collaborate with their lab partner and usually figure things out on campus. Your online students do not have those capabilities. Almost always they're conducting these experiments in isolation of other students and their instructor. So it's imperative that we are preemptive in planning for any questions that the student may have and addressing them with how we write the steps and with the photographs that we provide within the step-by-step procedures. Now, throughout the steps, students are referred to a series of data tables or other data collection devices. And these data tables and data collection cells are always provided within the lab manual platform. And they all exist in the lower right-hand corner labeled data. When the student clicks this, a data table maximizes on their computing device's screen and makes it very easy for them to input their observations and measurements. Any input that a student makes is saved every 3rd second. So if a student should lose connectivity for any reason, they do not lose their work. Furthermore, a student doesn't have to worry about forgetting to hit a save button in order to submit their data or photographs or whatever they're being required to submit as evidence of their experiment. So it is always saved on the platform. Again, if a student loses connectivity, as soon as they reconnect and open back up the data tab, all of their data is there. It was never lost. This is also very important for your students that are using non-traditional desktop or laptop computing devices, meaning that are using tablets or other mobility systems. In those types of devices, students cannot easily have different types of software open at the same time, meaning they can't have a remote connection to a digital platform open and easily then access an ancillary file, such as a Word document or a PDF or something like that. So it's imperative that we integrate all the content that a student needs to not only perform their procedures, but also record their work all within the digital lab manual. Now, at the bottom of every procedure page, student is going to encounter a series of open-ended questions. And these questions are specifically designed for them to relate their data and their experimental outcomes to the concepts that are central to this particular lab. These are always short answer by nature. And again, it gives the student a great opportunity to demonstrate just how much they understand about their experimental results. Many labs exist as more than one experiment. In this particular instance, we see there are two sets of experimental procedures covered in the enzymes lab. Here's an example of those chemical pipettes that I just wanted to share with you briefly. Each of them contains 1 ml of chemical. The tips are hermetically sealed, and the student just uses a pair of scissors to cut off those tips. And then they can control how much chemical they disseminate into each of these wells within a well plate, 1 drop at a time through these dropper pipettes. Again, the bottom of the procedure page has a series of open-ended questions, and the student has easy access to the data tabs. Now, at the conclusion of the exercises, again included in the experimentation section, once everything is complete, the evaluation system is unlocked for the student. The evaluation section serves 2 purposes. One, it's a quick review of the learning objectives. So the student can review and reflect on everything they should have achieved by this point. And then most importantly, the student completes a final exam for this particular lab chapter. When a student begins the exam, they are being timed so the instructor can view the time, and they are also being prohibited from returning to the previous lab manual pages. So once a student begins this competency review, the only thing they can do is the competency review until they complete the activity. And this one you can see has 9 questions. These questions are always multiple choice or true false in format. We've seen examples of these types of questions already. And as the student answers each question, they advance then to the next. At the conclusion of the questions, the student hits finish, and then their answers are all locked in, and the student is provided with feedback. We've seen examples of the feedback already, so that the student is made aware of which questions they answered correctly and those that were incorrect. And here we see examples of that. And then lastly, the student has an opportunity to answer 1 or more extension questions. And extension questions are designed specifically for a student to apply the knowledge they've gained from conducting the hands-on procedures to address a new problem. So this is a knowledge application activity, not unlike your students would be expected to do in their professional lives. So if you want to know really how much your student understands, here they're being expected to apply that knowledge. Again, short answer in nature, and students can use as many characters as they want to provide those answers to you. So that concludes an overview of the student experience. I want to touch on 2 things really quickly. And that the student does have some tools. They can call up not only all their lessons for a course, as we saw on the landing page, but a student can also review all the materials that are going to be used throughout the teaching term and so throughout their lab manual. This is important for students at the beginning of the lab course, so they can review the student materials for every lab. So if there are some materials they don't already have on hand, they don't have to make multiple shopping trips to acquire those. Just the next time they go to the store, they can jot down a quick list and they'll be all set for the entire semester. It also gives the student an overview of everything that's in their lab kit and which labs it's going to be used in. And then lastly, in this tab, the student has access to any supplemental documents for their course. If they're going to be dissecting specimens, they'll find some documents we've housed on their course about how to dispose of those. In other disciplines, they might find some Q&A documents, like in microbiology, for example. Or in chemistry, you know, they might find reference or a printable version of a periodic table, so forth and so on. And then for courses where students are to use a virtual microscope, they'll find the link to that digital tool within the tool drop down, again, in the upper left-hand corner. So I'm going to take a quick breather. I'm sure you've had a lot of questions come through. It'll give our panelists time to answer those questions. And if there's any questions that are specific to the student experience that come through that need to be addressed by myself, I'll spend the next 2 or 3 minutes addressing those vocally. So everyone can get the same answers, and then we'll get started on the rest of the presentation today, which is only going to take about 15 minutes. But I want to spend the rest of our time today, once I start back up, looking at instructor tools that have been built into our online learning platform. And those tools are going to give you, the instructor, the ability to customize your course for the needs of your students. It's going to give you the ability to provide feedback to your students based on their questions through the gradebook tool. It's going to give you the opportunity to upload a variety of supplemental information for your students. And then also it's going to give you the ability to create unique content and integrate it within the existing lab manual that's been provided to your students. So we'll briefly cover all those things, but if there are any immediate questions that pertain specifically to students, I'll be happy to answer those right now.
Thank you, Dr. D. Yes, so we had a few questions come in asking about what happens if students make a mistake and as a result they need a replacement of any of the kit materials. What is the process in place for that?
Sure. Almost always, even though we provide very small volumes of chemicals and materials in general in the kits to assure their safety, almost always there's enough material, raw materials in the kit for the students to repeat the procedures. So meaning if they mess up the first time, they almost always have enough to try again. Now, there are a few exceptions. You know, obviously, if we were talking about a dissection and a student just really mangled something badly the first time around and they couldn't go back and do it again, then that would be an exception. But if we're talking about chemicals and reagents and things, liquids and things such as that, we're almost always going to have enough for at least 2 times in there. If students should do something on their end that destroys something that they would have needed to use again, like if they dropped and broke a beaker that was designed, a glass beaker that was designed to be used in more than one experiment, or anything along those lines, or if they receive something that's damaged, they always just need to call us and we can replace anything. So it's not a problem. But ideally, aside from students destroying or breaking things, they should be able to repeat procedures at least one additional time.
Great, thank you. And then we had a couple questions pertaining to the type of microscope that is provided to students. Is it compound, internal light source?
Yes, great question, thank you. First of all, I want to mention that all of our curriculum is microscope agnostic, simply meaning there's only one version of every lab that requires microscopy. And we write the content in a way that whether a student is using a physical microscope or a virtual microscope, it's complete enough for them to be successful. Now, speaking of actual real microscopes, we offer a 600 power compound light microscope with an integrated light source, illuminator, that's battery powered on the illuminator. So So students that don't already own a microscope or have access to one, and they're taking a course where the instructor has specified that they use a real microscope, then we do have that option. And all of our curriculum is designed around a compound light microscope. Now, if a student's using a microscope that they already have or that they have access to, our minimum magnification, total magnification requirements for viewing prepared slides is 400 power. So they need to have a microscope that's at least that powerful. Again, the model that we offer as an option for students is 600 power, maximum magnification for biology courses. But again, a student can get by with a 400 power magnification if they already have a microscope.
Perfect. All right. Thank you. I think that covers it for the questions that haven't been addressed individually. So I think we're ready to move on to the instructor part.
Let's do that. Let's get into the instructor side of things. So now we're in instructor view for the same course. You'll notice the first difference is, as an instructor, I have the ability to type messages to students in real time and publish those by hitting save. And now, if I return to student view, just to toggle back, you'll see that all of my students have it again in real time. So I can immediately communicate within my course to my students on the landing page. So very easy for me to do. I also have the ability as an instructor to delete any old messages in real time for my class. The second thing I can do as an instructor to really match the flow of my lecture is I can rearrange the sequence of labs in the class. And again, this works in real time for my students. So the next time I toggle in student view, and i.e. to see what my students are currently encountering, they see the new order of labs. So again, this allows you to customize things. It matches now the flow of your lecture. And then the last tool you have here on the home page for students as an instructor is you can check or uncheck this sequential completion requirement. Now, the sequential completion requirement means, as we're looking at the screen right now, that a student would have to complete and submit the cellular respiration lab before they could begin the invertebrates lab. And you have control over that. You also have control, although I don't have the ability to show you right here, you also have the ability to hide lessons. So if you only wanted students to see only cellular respiration and not even see those other titles into some future date, you can control that as an instructor. So let's see how your view changes now within a given lab. So let's click on cellular respiration. We haven't looked at this one yet. And we'll click on exploration just as we did before. Now, you still have preview of everything that the student is doing. So you can still see all of their questions that they're answering, just like the student did. However, once we get to the content pages, things start to differ. You'll notice that as we scroll down this content page, we encounter these pulsing blue lines. Now, as an instructor, students don't see these, by the way. They're strictly for instructors. These are customization tools. It allows instructors to add content to existing labs. And that content can take the form of text, it can take the form of images. We get another blue bar here. And I can drag and drop either a PNG, a GIF, or a JPEG file, or it can take the form of a video. So if you had a video about a particular topic that's being covered in the background, you could upload that video directly onto the cloud platform, and it's there for your students to view. So again, every content page, an instructor has the ability to upload text, video, and supplemental images. Now, when you do that, anything that you've uploaded on the platform appears highlighted on the student's view of the page. So their attention automatically is focused on your content. Now, we don't restrict this content customization to just the exploration section. You can indeed, in the procedures, also upload supplemental content for your students in the same 3 forms. You can upload text, images, and videos. So let's say you've shot some videos of your on-campus students conducting certain procedures. You can share those videos or results or whatever you want to share with your online course with those students, again, by simply uploading that MP4 file as video directly onto the cloud, and then your students can view what their on-campus peers are doing. Now, when you click a data tab as an instructor, you see the ideal answer that your student will upload. So in this experiment, students are creating a graph of their results. They're also taking a photograph of, looks like the germination results of their seeds here before they put them in the respiration chambers. And you can see what an ideal student results look like there. And then here, you can see some raw data that the students were using to generate that graph. So unlike the student who just had blank panels, you actually see them with the results populated. So that's a quick overview of how you, the instructor, can not only change the sequence of labs in your course or hide them from students or change whether one gets completed before the other, but also, and very importantly, how you can upload supplemental content for your students in any of our existing lab manual chapters. Now, we also give you the ability as an instructor to add to the supplemental document files. So here are the same 2 files that the students saw that were already living on the platform for them, but you can drag and drop any other files you would like for the student. Now, why would you want to do a file here as opposed to make the content live within a lesson? Well, the main thing is on these particular files, students have the opportunity to download these files and store them on their computing device. So they can then easily print them or reference them at a later date. Whereas when the content is live within a lesson, the student doesn't have those same capabilities. We provide each instructor with all the answer guides for the labs in their course that you can download these and have a permanent copy of the correct student answers. But more importantly than that is we've designed a gradebook tool for you. So this allows you to grade your students in the course environment itself, and then very importantly, provide those students with feedback so they can review any notes you've made regarding their performance. So here we see the students enrolled in this demo course, and you'll notice that, you know, a lot of these students are celebrities. The pandemic has kind of required they go back to school since they can't film, can't record the music or anything like that right now. So don't be surprised with what students you're going to have as you send your class online. Here you'll see each student's progress as a snapshot as an instructor. Green circles means the student has submitted the work for that lesson, but that you, the instructor, have not assigned a numeric grade yet overall. White circles mean the student has not begun the lesson. Gray circles that we see for the first student here, Snoop, means that that student has started a lesson, but they've not finished. And then lastly, lessons with a number inside, meaning all grading is complete. And it looks like these students are all a bunch of superstars, literally, because they've all done great. Looks like they're all A students. Good for them. So what else can you do as an instructor? Well, you can determine the points for the auto-graded questions on the go. And ideally, you want to do this before your semester starts. So as I click on cellular respiration title, you'll see it has 3 test your knowledge activities that the students do before they get started. You can go in and assign a point value from as low as 0 to as high as you want. Now keep in mind those test your knowledge activities are formative assessments. You may not want to grade those, and that's okay, because they're really just designed to let a student know, make them aware of any knowledge gaps they have coming into that particular chapter. The exploration question time activities, or sorry, you may recall, are those questions at the bottom of each content page. You may want to grade those, and you can again assign as few or as many points to each of those questions as you choose. Now, once you've set the point value, your work is done on these interactive questions. The system grades them automatically. Then in the experimentation section, as you recall, there's a series of data tables and graphs and photo panels, and then those open-ended discussion questions. You can set up point values for those in advance, i.e. right now, or you can grade those as you go. It doesn't really matter what you do on that. And then finally, the competency review questions, just set a point value. These are those final exam questions, and the system will grade them automatically for you. So what does it look like when we grade a particular question? Well, let's click on a question here, and we will see. Here is our integrated answer key. So earlier I showed you could download answer keys, but here we provide you with answer keys integrated into the platform. And then here are all the students in my class now, and I can just click on a student and I can grade their results by clicking on then a particular chapter and a particular question within that chapter. I can leave the student notes that explains why I gave them the amount of credit that I did. And again, when a student then logs back onto their lab manual, they will see any comments that you have left for them in the grade book. So this cuts out a lot of work and time for you, in that you don't have to mark up a static document and then resubmit that to your student. Everything is digitized here. Everything's in real time. So very, very easy for you to communicate on the go with your students about their progress in your course. So what other tools have we made available to you? Well, the ultimate tool we've given you within the Lab Manual platform is called Custom Lesson Authoring. Now, I know you've probably been looking at our content and online through that link that you received at the beginning of the webinar today, and you may have seen, or may rather have not seen, one or more topics that you always teach on campus and that you really had planned on teaching for your online version of the course. Well, rest assured, if there's something that we don't currently support, we don't want that to prohibit you from taking your class online. And you indeed can use all of our authoring tools that myself and the rest of our team have available when we create content, to create your own unique content and present it to your student within the existing Lab Manual platform. So if there is that unique topic out there that you always cover and you want to provide it for your students and upload some videos to go with it and some questions and all of that, you have the liberty to do so. Conversely, if you want to use the Custom Lesson Authoring tool to simply create an overall end of semester practical auto-graded exam for every student in the class, you can do that. Any content that you create, you can drag and drop and disperse it in any order with the existing titles that already exist in your adopted lab manual. So you have the ultimate flexibility and freedom then to create a custom course that really meets you and your students' needs. And then very importantly, we've given you tools to integrate our lab manual platform within your existing learning management environment. So whether you're using Blackboard or Canvas or any other major learning management systems, you can actually integrate not only our entire platform, but individual lab manual chapters within your learning management environment, so that your students then really do encounter the photosynthesis lab immediately following the photosynthesis lecture that they're encountering in your learning management platform. And to do that, you just incorporate the efforts of your IT department. We have complete user guides and how-tos available to them for how to do this, and we are available to support them online or in various GoToMeeting type Zoom sessions to assist them in any level of integration that they desire. But I just want to let you know that if you want to integrate our lab manual within your pre-existing course content within your learning management system, that is very much an option for you. If conversely, all you want to do is to provide your students with a link on where to go to use their lab manual within the cloud environment, you can do that too. So you can choose to integrate, or you can choose to just give your students a link. Totally up to you, but we're here to support you in either way. And then lastly, I know I always get a question, so I'll just address it now about ADA compliancy. And I want to let you know that our content is screen reader compliant. Any videos that we upload onto the platform are closed captioned. Any images, so all those graphs and photos and diagrams that we upload are descriptive text provided in addition to those images to, again, meet ADA compliancy. Any technology tools that your students are expected to use, such as those drag and drop activity questions or the multiple choice questions or anything like that, are keyboard compatible. So students don't have to use a mouse to do those things. So we have taken all of these things into account and designed the platform accordingly. I know your school probably has a variety of regulations they have to follow regarding that, and we want to let you know we are very much aware of that and accommodating along those lines. We're almost out of time. I want to answer questions. Again, if there's enough interest in seeing the vScope, we'll look at it very, very quickly, but I don't want people who have a hard stop at our predefined limit of an hour today to mess out on a question they really wanted to answer in lieu of vScope. So if there are any questions, I'll turn this back over to you and our moderators and make sure we get those answered.
Thank you so much, Dr. D. Yes, we did have, we had a couple more questions come through. One was pertaining to the calculations. Are there calculations students are required to do throughout or after a lab?
It really depends on the lab itself. So yeah, in some instances, students are doing calculations. We tend to see those more in the disciplines of chemistry and physics than we do in, say, A&P and bio and micro and things such as that. But certainly, if students are collecting numeric data, as we saw in that cellular respiration lab, then they are going to be expected to, at a very minimum, to graphically analyze that data. As we saw that it had a graphic upload panel where a student did create a graph and upload an image of it. So yeah, it just depends on the type of data that a student's collecting. I mean, obviously, if we're in a lab where we're dissecting a fish or something like that, then we don't really have numeric data and we're not making calculations at that point in time. So again, if the topic dictates that type of work from the student, then yes, then they are going to be expected to do it.
Okay. All right. Thank you. And we do have a ton of interest in seeing the V-scope from the attendees. So do you think we have enough time to quickly show?
Oh, absolutely. So let's go back into that. We will take a quick look at the V-scope, but I am very cognizant of everyone's time budget. So this is our V-scope landing page, and we modeled the V-scope after obviously an actual working microscope. All of the slides that the V-scope uses are identical to glass prepared slides that we ship in kits for students that are using real microscopes. So there's no dichotomy as far as the slides that are being used or how students are expected to manipulate the microscope. Now, yes, students are expected to manipulate the virtual microscope. This is not merely a collection of images that students click on. This is a functioning microscope. Now, we had that prerequisite lab that we saw at the beginning today, but students are also provided with a quick checklist of how to use this should they have forgotten from their original tutorial. So just like any microscope, before a student can use it, they need to lower the stage. Again, these controls are keyboard accessible if required, and there's a how-to user guide by clicking this keyboard accessibility tab. I'm using a mouse though right now. We have to turn on an illuminator, or obviously we won't see any of the images that are produced by our slides. Then we actually select the slide and put that on our microscope. I'll just click something random here. You'll notice that as soon as a student puts the slide on the microscope, they do not have a usable image, not unlike if we were putting a real slide on the stage of a microscope. So we're going to need to center that image. We're going to need to raise the stage. We're going to need to focus and pan and do everything that a real microscope would do. And again, we have all the controls here that are animated on this virtual microscope interface that an individual using a real microscope would have access to. And let's see if we can't get this into fine focus. There's a little better focus, still kind of blurry. There we go. Well, it looks pretty close anyway. I can change the objective lenses. And then I can always refocus my image as needed. And I can also recenter an image or pan for new features under any level of magnification. When I'm, as a user, happy with the image that's showing within the field of my view, I can then download it. It's going to download as a JPEG file, as you see here, and I can then drag and drop that image into a photo panel within my digital lab manual. So if a student's using a real microscope, we provide them with a tutorial of how to use a cell phone camera in the barrel of their microscope to capture an image of their focus slide. For the V-scope users, once student has to have everything in focus, they just click download image. In either instance, the students are then having an image file that they need to upload into their lab manual program in order to submit their work. And in this case, the student would just drag and drop this JPEG file into that photo panel in their lab manual. So very straightforward. We have an extensive slide library. Now, our library is unique to our curriculum, but as you can see, there are well in excess of 100 slides in here. Every slide has the ability to pan around the slide image and to focus and to view that slide through all three objective lenses. So again, whether your student is using a real microscope or this virtual microscope is your choice as an educator, but every student in your course has it's subjected to the choice you make. So you can't have some students using a V-scope and some a real microscope. But that's something you get to choose as an instructor when you adopt a kit for your course. If you have any questions on the V-scope, I'm happy to answer them. I know we're already out of time, so I'll make it as quick as possible.
All right, perfect. Yes, we have a couple questions coming in. Does the V-scope have slides that represent a hemocytometer? I'm so sorry if I'm mispronouncing this. Hemocytometer for cell counting practice using white and blue live cell counting?
You know, I don't have a lab on an accounting lab that you're describing like that in the V-scope, mainly due to cost, not cost in the V-scope. That would not be prohibited for me as a curriculum provider. But to provide students with a real counting chamber, that would be using a real microscope. That is cost prohibitive. If you've ever priced out those counting chambers, they're not inexpensive. Yeah, if we can cost average them for a group of 30 students in a face-to-face lab, maybe not so bad. But when you consider your online students are conducting these experiments on their own, so everything they need has to be in their kit. There's just some items that I can't, from a cost consciousness standpoint, put in the physical kit for students. And counting chambers, at least ones that are of high quality, are not really an option at that point in time. But thank you for that question. And if we only did virtual things, then certainly I could do that.
All right, perfect. And then one more, because I know we are running a bit over time. So one more question regarding the V-scope is there an ocular micrometer within the microscope?
I do have a micrometer slide, yes. So I think it's labeled field of view, pretty sure on there. Here we go. But yes, to that question, because that is an expected activity for even students using a real microscope, is to calculate their field of view at all of their objective lenses. And so to accommodate that need for students using the V-scope, we have a field of view slide, which is a micrometer slide. Yeah, field of view. So yeah, it's in there. So I'd have to focus on it and everything, but it is a little ruler slide, so not a problem. Thank you for that question.
Perfect. Thank you so much, Dr. D, for all your expertise and your presentation today. That does it for us today. Thank you, everyone, for your amazing questions, for your engagement. We really appreciate it. We will be following up with that email that includes the webinar recording, so you can share with your colleagues and view anytime. Thank you again, and have a wonderful day. Take care.